Hello everyone. In the last video, we have started the tools. In the tool, we have started the enzymes. So the first enzyme already we have completed. That are the restriction enzymes we have completed. After that, the third type of enzymes we are coming. That are the DNA ligase. Always remember what are the DNA ligase enzymes, right? So ligase enzymes, they are adding enzymes. Okay, that means two DNA strands or the two DNA fragments can be attached to each other by this enzyme, DNA ligase enzyme. Suppose you have one cloning vector. With the cloning vector, if you want to add one foreign gene to make one recombinant DNA or in order to form one rDNA, then this gene fragment will not going to attach with each other by normal process. Why? Because in this enzymes, you can see this enzyme will help in the formation of the phosphodiester bond. See, first thing that in this gene fragment, the phosphate group is removed, right? And this cloning vector, it is also not have the bond formation that can be possible. So for your easy understanding, I have drawn one diagram. This is one DNA fragment. Suppose this is one DNA fragment. So what happened if suppose two DNA fragment, this is complete one DNA fragment. If you need to break it, then what you need the enzyme that are the removal, which help in remove the phosphate group, that is the alkaline phosphatase. Okay, so this alkaline phosphatase will remove the phosphate. So if suppose this phosphate group is removed, then what will happen, that this DNA fragment, see this phosphate group and this phosphate groups are removed, then this segment will be a different segment and this segment will be a different segment. After that, even if they are present together, they are close to each other, then also they will not able to form any bonds. Why? The reason is they don't have the phosphate bond. So alkaline phosphatase, this is actually a type of enzyme which help in removing the phosphate group from the 5' end, right? So what is the 5' end now? This is the structure. This is O, and here this is the first, second, third, fourth and the fifth carbon atom. Okay, so in the fifth carbon atom we have the phosphate group, and in the third carbon atom we are going to have the hydroxyl group. So here what happened, we have the OH on the 3' and here on the 5' end what they have the phosphate. So what actually alkaline phosphatase will do? Alkaline phosphatase will remove the phosphate group of the 5' end of the DNA fragment. So that's why we can able to get a completely two separate strands of DNA fragment. That's why we are able to cut the DNA segments. And after that, if you remove the phosphate, then it is not possible that once again they can form the bond. So that's why if you want to make or if you want to ligate, if you want to join two DNA fragments, then what you have to do? You have to add the phosphate group. Then only it is possible that once again we can able to form a complete DNA segment or we can form the rDNA. So from the 5' end, what you have to do? You have to add the phosphate group, right? So what is the DNA ligase? This is also known as genetic gum. Remember, it is going to add the DNA fragments. It joins the DNA segments by formation of the phosphodiester bond, right? So how the phosphodiester will be formed? That it will add the phosphate, okay, it will add the phosphate here and it will add the phosphate here, okay, in the two strands. So that's why what will happen, a bond, that is the phosphodiester bond. So this bond is the phosphodiester bond. After addition of the phosphate, the phosphodiester bond can be formed, and after that the two DNA segments can be joined to each other in order to form one rDNA, recombinant DNA. Right? So this is the DNA ligase enzyme. So DNA ligase, the most common DNA ligase is extracted from actually the T4 organism. So T4 DNA ligase is the most common DNA ligase enzyme that is isolated by one famous Indian scientist, Indian molecular biologist. He was H.G. Khurana or Hargobind Khurana, which is actually the first scientist to synthesize the RNA in laboratory condition. So he is the scientist. You remember that name. He is a very prominent name in the molecular biology field. He is an Indian scientist, Hargobind Khurana. He for the first time synthesized the RNA in the laboratory condition, not from any other organism, by just normal synthetic process. In the laboratory condition, he synthesized the RNA, the genetic material RNA. Now this common example of DNA ligase, that is the T4 DNA ligase, which is also it is also that one enzyme, that is the DNA ligase enzyme extracted from the same scientist, by the same scientist, that is Hargobind Khurana. Remember that. And the second enzyme, already I have told, this DNA ligase will add the phosphate and alkaline phosphatase will remove the phosphate. So it removes the phosphate group from the 5' end, and that's why we are able to get the two complete separate DNA strands. Okay. Next one, we have another group of enzymes. So these enzymes are going to be the last enzyme. What are they? They are the synthesizing enzyme. So they are helping in the formation of the different strands, DNA, RNA from the different genetic material, from the different nucleotide. So what are the synthesizing enzymes? See, DNA polymerase, then RNA polymerase, reverse transcriptase are used for different technologies. Okay, so what the DNA polymerase is used? DNA polymerase is used for the formation of the DNA, new DNA segments. RNA polymerase will help in the synthesis of the RNA segments. Next one, reverse transcriptase. Reverse transcriptase enzyme is used for the synthesis of the DNA from the genetic material RNA. So from the RNA, DNA can be synthesized, which is actually formed by whom? This is formed by actually the life cycle of HIV. You already know reverse transcriptase was carried by the HIV virus, where the genetic material is RNA, to incorporate into the host body, that means our body, inside our nucleus, they have to incorporate. So how they will able to incorporate? By the reverse transcriptase enzyme. So with the help of the reverse transcriptase, the RNA can synthesize the DNA. So this reverse transcriptase is a very much useful enzyme for different biotechnological approaches. So these are the different synthesizing enzymes. So we have completed the group of enzymes, where we have completed that cell membrane breaking enzymes, that are the cellulase, protease, lipase, chitinase. Those group of enzymes we have completed. Second one, the restriction enzyme, nuclease enzyme, we have completed. Third group of enzymes, they are the DNA ligase, which is going to add the genetic materials and form one complete DNA sequence or complete DNA segments that helps in formation of rDNA, that is the DNA ligase. Next one, alkaline phosphatase, which is also known as AP, which is helping in breaking the two DNA segments by removing the phosphate group. Next one, synthesizing enzyme, which is helping in the formation of DNA from DNA. RNA polymerase, which is helping in formation of RNA from RNA. Next one, reverse transcriptase, which is helping in the formation of DNA from RNA. Then normal transcriptase enzyme that will help in the formation of RNA from DNA. So this way lots of synthesizing enzymes are there. So we have completed the tools, that is enzyme. Hope you have understood. So in the book all the enzymes are not here, but then also restriction endonuclease, that group of enzyme that is very much important. And along with that, also remember the DNA ligase enzyme, they are also important. And another group of enzyme, the first group of enzyme we have discussed, that are the breaking or the cell breaking enzymes. They are the, for suppose bacterial cell, what enzyme is used, for suppose plant cell, for fungal cell, what type of enzymes are used. So those group of enzymes will be needed. So these are the important tools for that tool we have discussed, that are the enzyme. So that's all. So that is all about the enzymes. We are going to end this video now because the different other next tool, that is the vectors, we are going to start in the next video. So that's all about the enzymes. We are going to complete the video here only so that we can able to start the new part in the next tool, vector, in the next video. So that's all about the video. I hope you understand. Thank you.